microscope slide of the retina and things like that. So we're going to spend some time this morning talking about the retina. Remember, I'll show you another slide really quickly. If you remember, back up just a little bit. If you remember, just to use this diagram really quickly, the eyeball itself consists of 3 layers. There's that outer fibrous layer, the sclera and the cornea. There's that middle vascular layer that we have right here. This would be what we call the choroid of the eye. And then, of course, there's this inner neural layer that is the retina. And that's where we're at right now. So with that said, let's go ahead and kind of dive in and then take a look at the retina. Before we start, does anybody have any questions from last week? No? Okay. Cool. So I'm going to assume everything was perfectly clear. That's awesome. That's great. All right. So the retina is this neural layer. If you could dissect the retina out of your eye, you'd see that it's this layer of tissue that lays over the back of the inside of the eyeball, right? The best way to sort of think about the eyeball is the fact that it is a hollow ball, like a basketball. And imagine the inner part of that basketball is coated by this layer called the retina. If we were doing an eye dissection, you'd find that retina is kind of analogous to like a wet piece of tissue paper just spread over the inside of the eye. We're talking about something that's just about as thick and also just about as delicate, right? It's an exceptionally fragile layer that's kind of spread over the back of the inside of the eye, and simply just because it's not very thick, right? It's a really, really thin layer. So when we do eye dissections and things like that, it's really, really easy to inadvertently tear it and things like that. In fact, with eye trauma, it can get torn, it can fall off of the back of the eye and things like that. We call that a detached retina when that happens. It is held in place partially by the fluid that fills the eye. There's this fluid called the vitreous humor that pushes against the retina and helps to hold it on to the back of the eye, the choroid, and it helps to sort of keep that attachment. At the beginning of the lecture on the eye last week, I mentioned that the eye itself is just jam-packed full of sensory receptors. I mean, we're talking millions and millions and millions of sensory receptors that we have in the eye, and they're all found in the retina, okay, at least for the most part. So let's go ahead and take a look at the structure of the retina. So this is a cross-section of the retina, and the eyeball is like right here. So the front of the eye is right here. This is where the cornea and the cornea and the lens are at. And so if light were coming in, it would be coming in this direction right here. Okay, so it's kind of like the back of the eye that we're looking at right here. There are multiple layers of cells, and despite the fact that this is a really thin layer, there are multiple layers of cells that make it up. And what happens is that when light comes into the retina, it travels through the entire structure. Again, we're talking about a wet piece of tissue paper here, right? It's not hard for light to get through that, just like it wouldn't be for tissue paper either. We don't use tissue paper for blackout curtains for a reason. But nevertheless, we have the light that kind of travels through the retina in this direction. Now, in order to be able to see and form an image, we need our photoreceptor cells to be excited in a really logical way. If light were to get into our eye and start bouncing all around, then all of those photoreceptor cells would be excited, and we wouldn't actually be able to form an image of whatever it is that we're looking at. So what we need to do is we need to find a way, or we need to have a way to prevent light from bouncing around inside of the eye. And we've done that by evolving a fairly darkly colored inner layer of the retina. well as behind it. We talked about last week how the choroid is this really darkly colored layer of tissue. Those models that we have in lab, I think, show that pretty well. It's this kind of dark brown pigmented layer. And what that does is it works to absorb light. So when light hits it, right, light kind of comes in from this direction. When light hits the back of the retina as well as the choroid, which would be behind it, what that does is it prevents the light from bouncing all over the place and scattering, right? When light hits something like a mirror, it tends to scatter. But when light hits something dark like that, it tends to either get absorbed or it tends not to at least bounce, which is kind of the idea. To sort of aid that, the very back of the retina has a layer of pigmented cells as well. You can see this first layer of cells in the retina is that pigmented layer. All these little granules are melanin that are responsible for helping to pigment the retina and absorb light. Okay? So the pigmented layer of the retina melanin. You guys probably remember from 50A, melanin is the same pigment that we find in our skin. It's probably our biggest contributor to skin color and things like that. So we have a lot of melanin in this layer. It's going to be really obvious under the microscope later today when we take a look at this, that we have this heavily pigmented layer here towards the back of our retina. Okay. Now, despite the fact that this is meant to stop light from scattering, it doesn't stop it from bouncing back. So what happens is light kind of comes in here like this, and then it's going to turn around and it's going to be reflected or bounced off of it in almost the same direction that it came. Okay. This is when our photoreceptor cells become important. All right, so right next to the pigmented layer, we have our layer of photoreceptor cells. Okay? So this right here is our layer of photoreceptor cells. We've got two different types of photoreceptor cells. We have what we call rods and we have cones. These are probably something that you've heard of before. I feel like we talked about the eye a little bit in most anatomy classes, going back to high school or even middle school. So we have photoreceptor cells, we have rods, we have cones. Depending on how good our slides are today, we might be able to differentiate the two. Some of our slides are pretty good, some of them aren't so good, and our magnification is like almost just quite there, right? But when you look at diagrams like this, even though this is kind of an artist's rendition of this, you'll notice that, that there's a pretty distinct difference in the shape of these cells, and they are named based on their shape. You'll notice that the rods that we have right here have a very rod-like appearance, whereas the cones, the ends of those, have a very cone-like appearance, right? It's much more triangular in terms of its appearance. There's definitely a difference in the shape of these cells, and that is how they get their names. On this diagram, at least, I always feel like the cones look like little squid, right? I don't know if that'll help you remember anything or not, but they sure are cute. Nevertheless, we have our rods and our cones. Besides their shape, what else do you notice about the rods and the cones? Looking at that layer, do you notice anything about the rods and the cones? Looking at what's been drawn there, is there anything you can notice? Okay, so the cones connect to more than one bipolar neuron. They certainly do. Our next layer is the bipolar layer. These are our bipolar neurons, right? I've been telling you guys that they are only found in the eye. This is where we find it. Now's their moment. And you will notice that the rods connect to more than one, right, of these guys. So for instance, we have this bipolar neuron right here connected to 2 rods. We have this particular one connected to 3 rods, whereas the cones only connect to a single bipolar neuron. They don't connect to more than one. Or I guess, let me say that differently. Bipolar neurons connect to more than one rod, but they only connect to one cone. You can see that here. That's a good observation. Yeah. What else?
The ratio of rods to cones.
That's another big one, right? The ratio of rods to cones. In this diagram, what are there more of? Way more rods, right? We do have way more rods than we have cones, okay? We'll come back to that in a little while, but just kind of tuck that into your memory banks for the time being, that we have way more rods than we have cones. Okay. We'll talk a little bit more about some of the anatomy of the retina here in just a couple of minutes. But let's go on to some vision-related stuff. I have more to say about the retina, but that's a really good start. Actually, let me finish. Go back. I changed my mind. Just really quickly, just to sort of wrap this up. So what happens is these photoreceptor cells are going to be stimulated in the presence of light. Well, that's kind of what I want to lecture on today. They're going to be stimulated in the presence of light. When they are stimulated, they pass that information onto these bipolar neurons. Action potentials are started, and then the bipolar neurons synapse with these cells called ganglion cells that we see right here. And those ganglion cells have axons. They're also neurons, but they're really big cell bodies. They're called ganglion cells, and their axons are going to make up the optic nerve. Okay, remember, a nerve is a bundle of axons. The axons here are from those ganglion cells. Okay, so just to recap, light travels through the retina this way, all the way through the entire retina, and then as it's bounced back, it excites these photoreceptor cells, which excite the bipolar neurons, which excite the ganglion cells, which send messages to the brain, and your brain turns all of that into an image. Okay, so I do have a little bit more to say about the retina, but I think that gives us a pretty good overview of what I want to talk about today, which is kind of a little bit more physics-based than anatomy-based. So are there any questions? Any questions about what we've talked about so far this morning? Okay. So let me go up to my PowerPoint. Here, let's take a look at what we have. Okay, so I often will tell my classes that there are 3 things that kind of freak me out just a little bit. One of the things that freaks me out is space, because I just can't really comprehend something that doesn't end. That's really weird. Another thing that freaks me out is geologic time, because the whole idea of 1 billion years, or 2 billion years, or 3 billion years is really hard for me to wrap my mind around. I mean, 1 billion is a way bigger number than I think a lot of us give it credit for. So that just really freaks me out to sort of think about. Like, I just can't comprehend it, I just can't do it. The third thing that kind of weirds me out just a little bit is the scale of things that's happening inside of our own bodies. To think about the number of chemical reactions that are happening inside of your body literally right now is just overwhelming. I mean, it makes geologic time look easy to understand. And I think if there's another thing that I have a hard time sort of wrapping my mind around, if I were to add a 4th to that list, it would be vision and light. It's a little bit of an abstract concept, and the more I think about it, the more questions I think I have, which maybe is a good thing. But nevertheless, let's talk a little bit about vision and light. But bear with me because it's kind of an abstract sort of a concept, all right? So when you look at something, you see that it has colors, right? First of all, you see a shape, right? You see the shape of a person, you see the shape of a desk, you see the shape of a screen, and you see that there are colors on those things. Even if you were completely colorblind and you saw completely in monochrome, you'd see that there's differences in shades between things, right? For instance, right now I look out and I see blue shirts and I see pink shirts and I see red shirts and I see all sorts of stuff. Go Big Red, by the way. Yeah, Nebraska. I'm not a huge Nebraska fan, but they're next door to Wyoming, so I guess I have to be a fan. Most of my dad's family is from Nebraska. All right. So at any rate, we look out though and we see colors, right? We see colors on just about everything. The different colors of light that we see, and all light that we see, is all part of something called the electromagnetic spectrum. Okay, so we have the electromagnetic spectrum right here, which really is just a series of waves that exist in the universe, waves of energy. They differ really only in terms of something called the wavelength. All right. So if we have a wave, it can be any side of wave. It can be something on the electromagnetic spectrum. It could be a wave in the ocean. It could be any sort of wave. Waves have a couple of different characters to them, and one of those is the length of the wave. So if I've got a wave right here, something sort of repetitive like this, one of the things that I can do is I can measure the distance between peaks in that particular wave. So I can go from one peak to another peak, and I get something called the wavelength. Okay. Pretty easy, right? So I can have a wave like this, and I can have a wave like this. And you'll notice that the one on the bottom right here has a shorter wavelength than the one on the top. Does that make sense? All right, cool. Pretty straightforward. So the electromagnetic spectrum is looking at a series of waves of literally different wavelengths. For the most part, we aren't terribly aware of the electromagnetic spectrum around us. But on the long end of things, some of the waves with the longest wavelengths are upwards of a billion meters in length, right? So we're talking about really long wavelengths. Radio waves are something that exists on the longer end of things. Now we have transducers that help us capture certain radio waves and turn them into sound, which is what you listen to when you listen to the radio in your cars. You're picking up those radio waves and you're converting them to sound so that you can hear them. We'll talk a little bit more about sound waves and the longer end of the electromagnetic spectrum when we start to talk about the ear and hearing. Now, generally speaking, the shorter the wavelength, the more energy is packed into that wave. Okay. So when we shorten those wavelengths a little bit, we can get down to things like microwaves. Okay. And if you have a microwave at home, you are harnessing those microwaves to vibrate water molecules in your food so that you can heat your food up. That's essentially what a microwave does, is it puts out certain waves of a certain wavelength, and it uses those waves to vibrate the water molecules in your food, and you can heat up your food as a result of that. We have infrared waves, and then we run into something that we call the visible spectrum. Okay? The visible spectrum of light goes from about 700 nanometers down to about 400 nanometers. Now, if you guys remember your unit conversions from 50A, or if you have taken micro or maybe are currently taking micro, you guys are talking about unit conversions. Nano is a pretty small number, right? We're talking about really short little waves right here. And it exists from about 700 nanometers to about 400 nanometers. These are the wavelengths of light that excite the photoreceptors in our eyes. Okay? The photoreceptors in your eyes are the rods, and your cones are completely insensitive to microwaves, right? When you turn on the microwave to heat up your food, you don't see anything there at all. They aren't excited. But when you expose those photoreceptors to 700 down to about 400 nanometers, those wavelengths of light excite your photoreceptor cells. Okay. Going even shorter than that, we get into ultraviolet light, UV light. This is the type of light that is sort of a byproduct of the sun. This is enough energy in ultraviolet light to actually burn your skin, which is why you get sunburns. And then if we go down to even shorter wavelengths, really, really short, 10 to the negative 12 meters, we're talking about very, very short wavelengths here. We get into gamma radiation, and gamma rays can actually kill you if you're exposed to them without the proper protection, right? So those can be exceptionally damaging to our cells. So again, the shorter the wavelength, the more energy is packed into it, so much so that it can actually cause significant harm if we're not very careful about it. But what we're primarily interested in for vision is we're interested in what we call the visible section of that spectrum, or visible light. Okay. The really crazy thing, though, is that this particular range of wavelengths, the visible spectrum, isn't colored, right? There's nothing colored about any of this. These are just waves of energy that exist at different lengths, but there's no colors that are associated with these. And I think this is the thing that's really, really wacky, right? Is the fact that there is no color to any of this. I'm going to explain how we see colors, but there is no inherent color to any of this, okay? So how do we see color? Okay, it's solely determined by the light detecting cells that we have in our eyes. Okay, so let's go ahead and talk about this just a little bit. Okay, so we've got on the x-axis of all three of these graphs, we've got the visible spectrum, right? So we're going from about 700 nanometers down to about 400 nanometers. And then on the y-axis, we have what we call photoreceptor activation. So our photoreceptors being activated. Okay. And I also have some kind of things put in here, right? So we have red. It's what we call red light right here. It's about approximately 630 nanometers in terms of its wavelength. Yellow is right in here. Green is there, and then blue is right about there. Okay. So if we had only one type of photoreceptor cell, and we didn't have different kinds of photoreceptor cells, what we might see is we might see something like this. We might see that photoreceptor cell becomes especially active in sort of the middle of that visible wavelength of light, and that excitement kind of tapers off on both ends of it, so we see something like this. So if it's exposed to a wavelength of, say, 570 nanometers, what we see right about here becomes exceptionally excited. Okay, and we see whatever that color is or whatever that wavelength of light is being very, very bright. Okay, it's what you and I know as yellow, but again, there's no color to this, right? But look at this. Because this peak is sort of really wide right here, that single photoreceptor that you hypothetically gets just as excited for what we call green light. So you're not telling the difference at all between yellow and green. You're just seeing something that's really, really bright. Okay. You might see a little bit of a dimmer color for what you know as red. Okay, but you're not going to see it as red. It's still going to be some shade of gray. Okay, and the same thing is true for blue. And you're also not going to be able to tell the difference between these two because we're at the same level of excitement for both of these wavelengths of light. Does that make sense? Does that top pane make, are we seeing what's happening there? So just to kind of reiterate, you might see different shades or different brightnesses, but you're not going to be able to tell the difference between individual colors. Because for both of these, what we call green and what we call yellow, you're at the same level of excitement. So they're going to appear exactly the same. Red and blue are also going to appear exactly the same because they're at the same level of excitement or activation, which is our y-axis of our graph. Okay. So if you have only one type of photoreceptor cell, best you're getting is black and white vision. Okay, that's the very, very best that you're getting, right? You watch movies or TV shows or things like that in black and white. That's the best you're going to see if you have one type of photoreceptor cell. Okay. What if we had two types of photoreceptors? Okay. In the next graph, we have a photoreceptor that becomes maximally activated at a shorter wavelength than our other photoreceptor cell that becomes more activated at a slightly longer wavelength. Okay. This is hypothetical. We have more than this, but let's just pretend for a minute that this is what happens. When that occurs, this is when we get color vision. Okay. So here, we see that this photoreceptor cell becomes maximally activated at wavelengths at around 480 nanometers. Okay, and you'll notice that the other cell doesn't have any activation at that wavelength. If we look at this, you see that this guy does not overlap at all. All right. This particular one becomes maximally activated at around 600 nanometers. And you can see that we still have a little bit of trouble differentiating colors. Okay, we can't see that many different colors. We have red and yellow essentially producing the same color because of the way that this photoreceptor is activated. This is the same level of activation for both of these. Okay, but we see down here where we have green light, we see that there is some overlap. We see that this photoreceptor is slightly activated and this one is slightly activated. So that enables us to see an additional color in there as well. Okay. So because we have more photoreceptors being activated, it means that we have different signals going to our brain. Your brain is going to turn those signals into colors. Again, the light itself has no color. But when those wavelengths of light hit these photoreceptors, they become activated, they send messages to the brain, and your brain says, I see red when I look at this, or I see blue when I look at this. So the color that you see is how your brain interprets those action potentials. But again, the light itself has no actual inherent color. Isn't that wacky? What if we could see the world the way that it actually exists instead of the way that our brains interpret it to exist? That would be weird. Because there's no color. Our brains just interpret the color. That's a conversation for a different day, my friend. But isn't that crazy? Think about that. I mean, doesn't it blow your mind just a tiny bit that the world is only because your brain sees it that way? It's weird. Maybe it's just me. I don't know. But there we go. Okay. So if we have an additional photoreceptor, okay? If we have an additional photoreceptor, you'll see that we can separate out particular peaks, right? So for instance, where with two photoreceptors, we couldn't really tell the difference between yellow and red things. Now if we add a third photoreceptor in there, we can now get different signals to our brain, and we can actually start to tell the difference between those two wavelengths of light. We can start to parse out different colors as well, and we can see more colors. There's enough overlap here that it allows us to see all of the different colors that we see. In fact, we do have three different types of photoreceptors for color vision. We have cones that are particularly sensitive to red light, to green light, to blue light. You guys have heard of RGB before, right? And when they become stimulated in different ways, that's when we start to see different colors. And so all of the colors that you are responsible for seeing is because of three types of cones. What we call our red cones, our green cones, and our blue cones. And the overlap between them allows us to see all of the in-between color variations that we're aware of. You know, we mimic that all the time because that's kind of how our eyes are excited. We can mimic that as well. So if we go to the internet for a minute, and you can go to RGB, kind of RGB color picker of sorts, something along these lines. This is just you guys have probably all seen something like this before. But you'll notice that you have your little RGB right down here, right? So let's just say on the computer, if I only wanted to excite red, the RGB scale goes from 0 to 255. Not that you needed to know that necessarily, but I guess now you do. But if you went 255, and then 0, and 0, that would give you pure red, right? So the only thing that I'm exciting here in this particular instance is I've got a wavelength of light, so to speak, that excites only my red cones. I know that this example isn't perfect, but it kind of gives you an idea. But if that wavelength of light slides just a little bit over to the left, and it starts to excite my green cones just a little bit, let's go ahead and give this like a 75 or something like that. We see that the color changes just a little bit because now we're exciting more than one of our cones. And if it slides down even farther, or we're able to excite the other one just a little bit, let's go ahead and give that 30. You know, we see that it changes just a little bit yet again, right? So if we go back to our graph right here, we have light reflecting all of the things around us that hits our retinas, and different cells are excited in different ways, and that's what it allows us to interpret the entire spectrum of colors that we see. But we have to have more than one type of cone to be able to do that, because if we had only one type or even two types, the number of colors that we see would be drastically limited compared. Okay. So if we add in our rods, we see that we have 4 total types of photoreceptors. Our rods are not particularly good at detecting color, but they're good at detecting light itself. Rods are very sensitive to low levels of light. In fact, as the light dims, your rods become more and more and more active. We'll demonstrate that maybe a little bit in lab. Your cones are able to perceive color because they have different, what we call opsin proteins that are sensitive to different wavelengths of light. And then the brain, of course, interprets the relative activity of each cone type. So at the end of the day, this is what our sort of graph would look like. This kind of expands on what I had in the previous slide by adding rods into the end of that as well. Okay, so you and I have 3 different types of cones that allow us to perceive the world around us. To sort of piggyback on my philosophical rant just a minute ago, again, there's no color in light, so the only reason you see color in the world around you is because your brain is interpreting that. But to sort of piggyback on that, because it is just the number of cones that you have, and it's your brain interpreting that. If we added even more cones in, then we would theoretically be able to see more colors. Okay, and so there are some really good examples in the animal kingdom of animals that have a number of different types of cones. Butterflies were a really good example. They have 16 different types of cones. You have 3. They have 16 different types of cones. So butterflies conceivably can see colors that you and I don't even have a comprehension of, right? Because they're able to see those different colors. But the winner of all of them is this little guy right here. Does anybody know what this is? There's a number of different species of these, but what is it?
Mantis shrimp.
It's a mantis shrimp, okay? Mantis shrimp have 24 different cones. Yeah. So again, they're able to see colors that you and I have absolutely no comprehension of, that we don't have names for, that we don't even think about, because their cones are excited in such a way that their brain can parse out probably millions of more colors than you and I have the ability to even understand. They're also really colorful themselves, so there's got to be something there in terms of mating or something like that. Mantis shrimp are really, really cool. They're fairly common in most oceans around the world, just kind of as an aside. But they're really hard to keep in aquariums. Like, you may have never seen one of these before, even though you've been to aquariums. My kids have always liked aquariums, so we try to go when we can. But they're really hard to keep in captivity. And the reason for it is not because they're particularly sensitive, it's that they're incredibly destructive. They're violent little things, man. So you'll notice right here, this mantis shrimp has this is one of its appendages right here. It looks like a boxing bag, like a rhythm bag for boxing, right? But what it is that it's like it's encased with exoskeleton, so it's really, really hard. But what they have the ability to do is they have the ability to sort of kind of cock that back. And then they have a little latch in their exoskeleton that they can latch it into place. And then they use their muscle to pull really, really hard on it and build up a lot of tension. And then they can release that latch, and it flies forward with so much force that it can actually break aquarium glass. So what they'll do is they'll start going up to aquarium glass when they're in a captivity, and they'll just start banging on it until they shatter it. And they've actually shattered aquariums because there's so much force in that appendage that they're able to do that. Now, in the wild, what they do is they use it for eating snails. They'll go up to snails and they'll just hit them really hard, and they'll actually just shatter a snail shell, and then they can get in and they can eat the snail with no problems at all. But they're really remarkable little creatures.
They can punch so hard that they can knock off the claws off crabs.
Yes, yeah, they can knock claws off of crabs as a defensive thing. It's the fastest strike in the entire animal kingdom, right? It makes rattlesnakes look like super slow in terms of their feeding strikes. They're absolutely incredible. I don't know if anybody in here is a boater or not, who's ever had a boat, but one of the things that happens to boat propellers is that they go around the water so fast, they eventually wear out. Something called cavitation happens to boat propellers, where what happens is you get these little bubbles of gas on the surface of the propeller, and because the propeller is traveling so fast, that bubble ruptures with so much force it can actually wear away the metal on the propeller. So after time, you have to actually replace boat propellers because of cavitation. The propeller will literally wear itself down. These guys, on high-speed video footage, there's a woman, what's her name? Sheila Paddock at UC Berkeley has done work on mantis shrimp, and she's done super high-speed video footage of these guys hitting like snail shells. And she's actually captured cavitation on film. They hit them so hard, there's this really bright flash of light that occurs when they hit these snail shells. So they're actually wearing that feeding appendage down. They kind of constantly have to regenerate it. So really, really cool little animals. But what I think is cool is the fact that their eyes have so many different types of cones in them. They're really, really remarkable little things. Okay. So let's take just a minute and let's compare rod and cone vision. So as I said, rods are really, really sensitive to light. They're just not good at color, right? So because they are so sensitive, they become especially active in our night vision, okay? They also play a really important role in our peripheral vision as well. Like I said, black and white, but nighttime peripheral vision, things like that, they're very, very important. Somebody had noted a little while ago that a bipolar neuron may have more than one rod going to it. That's a really important observation. We call those pathways convergent pathways. And because those pathways do converge, it kind of confuses our bipolar neurons a little bit, and the image that's formed is a little fuzzy as a result of that, right? So it's not necessarily the clearest vision, but it does allow us to see shapes at the very least. So, one of the things that we can sort of think about is we can think about as the darker a room gets, we have our night vision sort of kicking into place, right? So if we were to do that really quickly. There we go. All right. So it's dark in here now. Don't worry about taking notes. It's not a big deal. But let's just think for a minute what's going to happen. Let's let our eyes adjust for a second, for one. So it's going to take a minute for your eyes to sort of adjust. But think about all of the colors on all of the shirts and things like that that people had on when they were in here, right? But now, as you look around the room, you see shapes, but we're not very good at seeing colors, right? Our cones have a really low sensitivity to light. So as a result of that, when a room is dark, like it is right now, we don't really have any cones that are being stimulated, at least not very heavily, right? So what happens is our vision becomes more and more and more monochromatic, right? It becomes more black and white. Our vision isn't as clear. You can definitely see shapes of things, especially as your eyes start to adjust. You can definitely see shapes of things, but the sharp edges start to go away just a little bit, and our vision becomes a little bit more fuzzy as a result of this, right? Think about walking around your house in the middle of the night, right? Things are definitely a lot fuzzier as you're walking around in the dark. It takes a little while for our night vision to sort of kick in. We're not going to give it the full time to be able to do that, but we can definitely start to see here that our vision is changing. But think about the whole idea of this, right? These gray, or these rods that are being primarily stimulated right now are giving us a fuzzy image of the world around us, and they do tend to be the most active in our peripheral vision, at least during the day. What advantage would it have to have really, good image sensitivity, movement sensitivity in your peripheral vision? I mean, let's think a little bit about maybe not our lives, but maybe the lives of our ancestors, right? Think about your ancestors out roaming the post-glacial plains of North America, right? Why might peripheral vision be important?
Protection.
Yeah, I mean, things want to eat you, right? And that's not a good thing. You don't want to end your day by being eaten. And predators are not dumb. They're not going to come right at you, right? They're going to try to ambush you. So if you have peripheral vision with high sensitivity to movement and shapes, it's definitely going to be an advantage for you to be able to survive and get through your day without being eaten, right? At night, it's really helpful, even though it may not be a sharp image, being able to see movement at night is going to be really, really helpful, because you might see a predator, or you might be more likely at least to see a predator sneaking up on you, because they're definitely relying on the cover of darkness to be able to surprise you. So being able to see those shapes in that movement, even though it's not a perfectly clear picture, is definitely going to help you to survive. So that's really, really important. Our cones are just going to give us a little bit more information about the world. So I'm going to unmute the screen right here, and then I'm also going to turn on the lights again. So get ready. And then we see here, and when we turn the lights back on, everything comes back to life. Our sharp vision comes back, those colors come back, and everything like that. Wake up! It wasn't that long. So our vision comes back, our colors come back, and we're back to that high resolution color vision that we're used to. All right. Okay. We'll come back to photoreceptors a little bit in lab today. You know, I wanted to talk a little bit about the visual pigments that make a lot of this work. How are we doing on time? We got a little bit of time. So your book goes into quite a bit of detail about this, but we're just going to kind of get a little bit of a brief overview for you here. So as far as the visual pigments go, the reason we're allowed to see is because we do have proteins in those photoreceptor cells that are really important. So one of the things that we're going to be talking about is something called retinal. Retinal is a light-absorbing molecule that combines with one of 4 opsin proteins to form visual pigments. It's synthesized from vitamin A, which is, I guess, why we always tell you you need vitamin A in your diet. These opsins consist of a couple of important things. First of all, we have something called rhodopsin. This is found only in rods, so rho and rod kind of sound similar. And then you have your cone opsins, your red, your blue, and your green. Okay, so those go into this thing called retinal. And these retinal isomers are different 3-dimensional sorts of shapes. Just like most proteins in our body, they have a very distinct 3-dimensional shape, and isomers are just sort of variations on the theme. You have a slight shift in the shape of it, and we call it an isomer. Okay. If you've had organic chemistry, you've definitely talked about isomers of different molecules because this is an important aspect of it. And typically, most things will change between one of 2 isomers. One's called a cis isomer, and the other is called a trans isomer. And it has to do with the arrangement of the atoms on the molecule. And when they change, we go between that cis and that trans isomer. Okay? So the 2 primary isomers of retinol that we're going to be dealing with, one is called cis retinol, it's called 11-cis retinol, and the other is called all-trans retinol. And really it just has to do with whether that molecule is bent or if it's kind of straight. Okay. The conversion of bent to straight depends on whether or not light is hitting it. Okay, so let's go ahead and kind of take a little bit of a look at what this might look like. So this is looking at your rods, but inside of our rods are these little tiny discs, and if we look inside of those, this is our plasma membrane right here. And this is our visual pigment embedded in that membrane. So you have the retinal right here. This is that little molecule that can change shape between cis and trans. And then you have opsin sort of surrounding it and holding it in place. This is kind of where I want to go. Okay. So we are going to see that when light sort of hits these guys, we can change between the 2 shapes. So this is a diagram sort of showing what's happening here. Everything where there's no light is obviously on this side, and everything where there's light is on this side. And so what happens is in the dark, what we're doing is we're essentially building up a whole bunch of this 11-cis retinal in here. But when light hits it, what happens is it's rapidly converted to the trans isomer, and then it has to slowly sort of regenerate itself. When that happens, we call that pigment bleaching, And if it all happens at once, which can definitely happen, you might see a very brief moment of blindness as a result of that, because you've built up all of this particular polymer or isomer of this, and then when you hit the light, it all changes shape at once. You might not actually be able to form an image. So that actually happens like when you go outside. I probably can't make the lights bright enough in here to do it, but if we're all adapted to our night vision, and then we suddenly turn on the lights for like a very brief moment, it gets very, very bright, and you're like, I can't see anything. That's because of that pigment bleaching. All of that cis isomer has been converted to trans isomer at once, and it just bleaches your vision out. And then as you slowly start to regenerate that cis isomer, that's when your vision will come back, and you're going to be able to actually stimulate different cells and things like that. So it's kind of an interesting process, but we're just going back and forth between that cis and that trans version of those guys. So that rhodopsin is sensitive enough that even a small amount of pigment bleaching can occur in the middle of the night. Just by looking at stars in the sky, we can have pigment bleaching that occurs. And then, of course, in bright light, bleaching occurs fast that rods are virtually non-functional. That's why you have that moment of blindness. Cones do respond very well to bright light, and as a result of that, we end up with our color vision. So light adaptation occurs when we move from darkness into bright light. We see that glare because both the rods and the cones are being strongly stimulated. We have that pigment bleaching that occurs. It takes a minute for our pupils to constrict, but once our pupils do constrict, our rod system effectively turns it off. That sensitivity of retinal decreases so that we can actually maintain some of that cis isomer, and then the cones and the neurons that are attached to them really rapidly adapt. So it takes a second. It's not an instantaneous thing for this to occur. As far as dark adaptation goes, moving into the darkness, we see blackness for a minute because our cones completely stop functioning. That bright light bleaches those rod pigments are still bleached out, so they have to adapt, and they have to build up their cis version of retinal, and the pupils also have to dilate. Full night vision doesn't occur for almost 20 to 30 minutes after we're in the dark. It takes quite a while for this process to occur, but nevertheless, it's an important. Okay. These two slides are really kind of the crux of all of this, dark adaptation and light adaptation. I need to pare down the slides that I have in here to be able to kind of help you guys with this, but these are the two that I think are the most important for us to focus on as far as testable material goes. It's a fairly straightforward thing. We could go into a lot more detail, but I don't want to overload us. So focus on these two slides primarily for dark adaptation and light adaptation, and I think that will get us there. All right. Okay. So it does get us almost to 9